Welcome to The Explainer. I'm absolutely thrilled to guide you through what is, quite frankly, a groundbreaking leap in how artificial intelligence is being used to design life-saving therapeutics. We're going to be looking at a brand new research paper detailing a system called AAMFM, and let me tell you, it represents a massive, game-changing shift in computational biology.
All right, let's just do a quick run-through of our roadmap today. We'll start with the core antibody design challenge, then we'll formally meet the AFM AI, break down how it adapts to the antigen and learns preferences, check out its state-of-the-art results, and then finally wrap up with what this all means for the future of therapeutics.
Okay, Section 1, the Antibody Design Challenge. We're starting our explainer with the fundamental problem of how our immune system actually recognizes threats. It's basically an incredibly complex, high-stakes biological matching game.
To really understand the magnitude of this breakthrough, I want you to imagine something. What if we didn't have to rely on those incredibly slow, labor-intensive lab screening processes, like massive antibody library panning, just to find a cure? What if instead we could literally mathematically generate the exact molecular key to lock down any virus, completely on demand? I mean, that right there is the absolute holy grail of therapeutic design.
So the crucial point here is that designing an antibody is all about perfecting its complementarity determining regions, or CDRs. You can think of an antibody as a key, right? And the disease, the antigen, is the lock. The CDRs are essentially the highly variable ridges and grooves that determine whether that key will actually turn the lock. The rest of the antibody is, well, basically just the handle. If you want to neutralize a very specific target, you have to engineer these CDRs with astonishing, almost atomic precision.
Now, I know what you might be thinking. Wait, don't we already have massively powerful AI protein models? And we do, but standard protein language models are kind of like trying to cut a physical key by only looking at a written description of a lock. They mostly operate in one dimension, just analyzing sequences of amino acids. They completely lack the crucial 3D geometry needed to make a functional physical fit. Because of this, when they try to design an antibody, they often spit out sequences that either don't fold properly in three-dimensional space, or they simply just don't bind to the specific target they were aimed at.
Which brings us to Section 2, Meet the AAMFM AI. Let's move to a completely new paradigm in computational biology that was built to solve this exact bottleneck.
So AAMFM. It stands for Antigen-specific Antibody Multimodal Foundation Model. Yeah, I know, it's quite a mouthful. But this architecture is so revolutionary because rather than just looking at flat sequences, AAMFM unifies sequence and structure into a single, shared mathematical space. It actually ingests both the physical shape of the antigen and the antibody framework to design something entirely custom.
To build this master locksmith, the AI essentially acts as an apprentice passing through three very distinct phases. First, it undergoes universal pre-training using a massive foundation model called ESM3, where it just learns the basic alphabet of all proteins. Next, it undergoes supervised fine-tuning on experimental antibody data to study specific structural interactions. And finally, the really cool part, it goes through preference alignment. This is a highly specialized phase where it actually masters the art of functional binding.
Moving right along to Section 3, Adapting to the Antigen. Let's explore exactly how this AI looks at the lock it's trying to open, which happens during that critical second phase of training.
Like we just established, standard models are pretty much flying blind. They know what an antibody sequence should generally look like, but they have no idea what it's actually attacking. AAMFM totally solves this by using a lightweight cross-modal adapter. And when I say lightweight, I mean it. This adapter is tiny, taking up only about 0.36% of the base model's parameters. But it seamlessly maps the physical geometry of the antigen directly into the AI's neural network.
What's really fascinating here is that this adapter allows the model to deeply understand the spatial relationship between the epitope on the antigen, the exact spot we want to target, and the CDRs it's generating. It literally projects the 3D atomic coordinates of the target right alongside the amino acid sequences. This means the AI can simultaneously optimize for both chemical makeup and 3D shape all at once.
Section 4. Teaching AI with preferences. But here's the catch. Knowing the shape of the lock just isn't enough. The AI has to know what makes a key truly good. And this brings us to what is arguably the most innovative part of the entire research paper. AFM is fine-tuned using a process called Calibrated Direct Preference Optimization, or Cal-DPO. Basically, the system takes an antigen and generates multiple candidate antibodies. Then it labels some as preferred and others as dispreferred.
We are effectively training the AI by constantly showing it the difference between a successful key and a totally failed one, forcing it to calibrate its own internal logic to favor designs that will actually work in physical reality.
Now you know how AI can sometimes, well, cheat? To prevent the AI from generating a shape that mathematically fits but would just immediately fall apart in a real human body, it has to satisfy a super strict dual threshold rule. First, the key must physically fit the lock with high confidence according to AlphaFold 3, which, as we all know, is basically the gold standard for predicting physical structures right now. But second, it must also score highly on antibody, meaning it actually looks like a natural, plausible biological molecule. It absolutely has to master both physics and biology to be labeled a success. No shortcuts allowed.
All right, Section 5, State-of-the-Art Results. Let's take a look at the actual numbers and see how all this brilliant theory translates into real, measurable scientific breakthroughs.
When you pit it against the most advanced current models out there, AAMFM just completely dominates. In pseudo-log likelihood, or PLL, where closer to 0 is better, it hits negative 0.87, easily beating the state-of-the-art ABEX model. But the real standout for me is the IPTM score, which specifically measures binding functionality. AAMFM sweeps the board here, proving for sure that its preference training pipeline creates designs that are vastly more functional and structurally sound than literally anything else available today.
And it gets even better. Even when we restrict the test to designing just the incredibly difficult CDRH3 region, which biologists consider the absolute most critical and highly variable part of the key, AAMFM maintains its massive lead. You always want the lowest possible RMSD, right? Because that measures structural deviation from nature. Well, AAMFM achieves a 2.62, which is significantly lower than the competitors. It's generating these hyper-specific loops that lock onto targets flawlessly.
You can actually see this difference visually, and it's striking. Look at the baseline ABEX model on one side. It clearly struggles with the interface. The generated CDR loop is a bit loose, kind of wobbly, and it's not quite perfectly conforming to the antigen structure above it. But then you look at our AFFM model on the other side. It generates a significantly tighter, highly functional structural fit. The loop perfectly nestles into the geometric pockets of the target. It's quite literally the difference between a key that jiggles loosely in the lock and one that turns smoothly on the first try.
Finally, Section 6, the future of therapeutics. Let's wrap up this explainer by zooming out and looking at what all of this means for the future of medicine.
As the authors perfectly state, we have officially crossed a massive threshold here. AI isn't just hallucinating random protein sequences anymore, hoping something sticks. It is actively engineering functional, plausible biology with real intent and precision, entirely guided by strict physical constraints.
Which leaves us with, honestly, a pretty staggering thought. If this multimodal framework works so flawlessly for antibodies, generating custom cures right there on a computer screen, well, we are standing at the absolute dawn of AI being able to engineer cures for nearly any protein-based dysfunction in the human body. The perfect key for absolutely any lock. Thank you so much for joining me on this explainer. I really hope this sparked your curiosity just as much as it did mine.